Welcome to the last part of the Info Day. No messing about, we'll get on with the next talk. It's on research and developments in myeloma. I couldn't think of a better person to talk to this subject than Karthik Ramasamy, who's a consultant hematologist in Oxford, but also runs a very busy clinical and laboratory research program there. So Karthik, over to you.
Thanks very much, Simon, and thanks to Myeloma UK to give me the opportunity. I've never been to a London Info Day. I often hear from my patients that London Info Day is the best, so thanks for having me here today. So the topic I'm going to cover is research and development. So to set the scene, I want to start with a show of hands, just to see how many of you are awake still, right? How many of you have ever participated in a clinical trial? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Wow, excellent. Well, give yourself a big hand because you're contributing to the continuing development of all the myeloma care that we're going to talk about. The second thing is, how many of you have signed a form to say that you're happy to have your bone marrow being researched on in a laboratory? Wow, double the number. Fantastic. Great. Okay, my last question. How many of you have ever been part of a panel advising NICE or working with Myeloma UK to advise NICE about access to new treatment? Okay, I can see about 2 or 3 hands. Okay, so there is a pitch slide to try and get more of you involved because we definitely need that soft influence that you can all give, but not just soft influence, but authentic stories, because a lot of NICE panels, as I was talking earlier, are chaired by myeloma doctors like me. So they understand the stories that people give. So we need to hear more of your stories if you were in part of a trial talking about that treatment when these are being assessed in the clinical practice. So at the outset, this is what my talk will cover. My intention is to run for about 40 minutes, and then we'll do a 20-minute Q&A on research and development. So the latest research or the latest understanding around how myeloma develops, a bit of this was covered earlier, but I will talk about that. We've got a few slides around that. The second thing is around everybody's myeloma is different and everybody's life story is different, and there are a lot of factors which influence patient outcomes. How do we actually personalize treatments, and what trials are happening to personalize those treatments? Because we now have many treatments in multiple myeloma. So that is something that I will touch upon. And then we'll move on to the new treatment approaches. And the 2 big new treatments are the bispecific antibodies and then the CAR T cells. So we're going to talk about those 2 and what is happening in that space. Someone rightly pointed out we're also doing trispecifics. That is true. If bispecifics are now licensed, we need to keep moving, right? So we moved to trispecifics now. And then we'll talk a little bit about the treatment approval process. Some of you are probably advanced in your thinking because I saw a few hands go up when you've been involved in the whole health technology appraisal process. But it'll be nice to get a leveled view of what that is. And if you've got any questions or comments about that, it will be good to hear. And finishing up about how you can all be involved in research. So the main messages are, in myeloma, the research continues to improve our understanding of how myeloma develops and progresses. I'm going to take you into a little bit of science as much as possible before losing you around what we are doing now around carbon dating, myeloma genome, and so on. So let's see how we land there. The next thing is around new treatments and treatment strategies that are being investigated, and why we're thinking about those treatments in that manner is what you need to think about. Not just about what treatments we're using, why we're using those treatments and how we're planning to use those treatments is how you need to question yourself when you see those things. And how do we actually end up personalizing treatment when we know that drug companies are going to continue to develop drugs for all myeloma, but we know that some people do well and some people do less well, and how do you actually use that messaging within clinical practice? And it's absolutely integral that patients and carers are continuing to be supportive of research. Otherwise, we wouldn't have done what we have done in myeloma over the last 20 years that I've been involved in. And it's amazing to see that how survival has significantly improved. When I started in myeloma in 2003, the number of people we would see who had survived even 5 years, 10 years was very, very minimal. But they were there. It's not that they were zero. It's very important that people don't recognize that. When I pulled data, even in 2003, there were some 10, 20 year survivors even then, but they were 1%, 2%. But now they are 40, 50% in some segments. So there's huge advances that is happening. And really, we have to reflect on patient preferences. So one message I want to land with all of you is patients and carers have to drive the research agenda. A lot of what has happened up until now is very much driven by pharma for the right reasons, because they had better drugs, they had to bring it in, and then they make a difference. But we've got to a place where things have now, there are exciting drugs, but they have to be placed in the right manner. So unless patients and carers get more involved in driving the research agenda, all what people will be doing is to just find more drugs, and how to get them more upstream is the very simple thinking that will continue to happen. If you want to change the dialogue, you've got to get involved. You need to find ways of getting involved, because there will be questions that you want answers for. They won't naturally happen. The doctors and the researchers are not going to think about it the same way that you're thinking. And unless you bring those questions to the table, we will be unable to take that forward. So it's very important that you understand. You've got to drive that. And the vehicle really is Myeloma UK and the Patient Carer Research Panel. It is your opportunity to continue to influence the questions that we ask. And the good thing is, not just Myeloma UK, all cancer charities in the UK and NIH welcome. And if you look at any other medical research charities, they want patient voice more involved in driving the research. So it's critically important you take home that message, as well as spread that message around to people who you're meeting. So these are the different elements that are being researched in the myeloma segment. And there are people who are laboratory researchers who sit in the lab and continue to think about who gets myeloma and why. I think someone ask the question as to whether there's a myeloma cell in the bone marrow or outside the bone marrow. So those type of very, very basic questions people are thinking about. Is it in the bone marrow it starts, or does it start in a lymph gland or a tonsil, or all these very, very basic questions. People are asking those right now in laboratories, because I gave a talk in Korea about 6 weeks back, and there was an 8-year-old with myeloma. And we were talking about what dose of lenalidomide to give to that 8-year-old girl, is what it is. So if that can happen, then we need to continue to understand how myeloma develops. The second thing is, thanks to Myeloma UK and thanks to a number of international research communities, we are increasingly focusing on the precursor conditions. Our focus rightly was in active myeloma and how do we get the right drugs to keep people living longer. We've spent a bulk of our time in that space, and it's important that we start to move ourselves upstream and think about, well, why did we get to the active myeloma in the first place? And we know that there are a lot more people with that earlier stages, and how do we prevent the problem happening for those people who are right now in a Piccadilly line or district line, not knowing that they've got smoldering myeloma, and how do we actually stop them from getting myeloma in the next 2 to 3 years is what we need to kind of think about. If we don't make that investment, then we will be continuing to kind of struggle and think about just purely improving survival for myeloma. So precursor conditions and how they develop, and lots of research on patient preferences and health services, treatment options, and then stratified medicine as I can come across. Okay, so how does myeloma develop and progress? So let's get some basic facts across, because then you could understand the basic facts and then start to think about, well, how do we actually, get deeper into it, and what type of research would we do to get to that question as to who gets myeloma and why did I get myeloma, okay? So these are the facts for you on this slide, okay? So older the person, more common is myeloma. So, that's one fact, okay? And we were very concerned when we first put this fact out that this was only common for, Western population, but we now have data in all different ethnicities, and this is a factual proof that older you are, higher your chance of getting myeloma. Second is more common in men. And interestingly, more common in men with active myeloma, okay? But even more common when you think about smoldering myeloma. It's almost 1.5:1 for smoldering myeloma. So then you may be sitting there wondering, well, if it's 1.5:1 for smoldering myeloma, why is it 1.2:1 with active myeloma? Well, half the men never bothered to get tested, so they're probably, dare I say, dying with a degree of active myeloma whilst they're dying, is what one hypothesis is, okay? Or maybe men have some change in their immune system, which is why they can stay smoldering before changing to active. So a lot of these basic fundamental thinking starts to come to your head as to why that may be the case. And those are all driven by these basic observations. Third, much more common in people with black ethnicity and much less common in people with Asian origin. I know people don't like the word Asian because it brings a lot of populations together, but essentially as you go down to the eastern part of the world, you see much less myeloma, is what you see. I gave a lecture to Indians about a week back, and they said the number of people they see over 75 is almost zero, because the life expectancy is low and they don't get there, and there are very few people they actually see over 75, whereas 45% of people diagnosed with myeloma in this country are over 75. So you see the differences, right? And that's why this personalization of care is even more important. It's not just finding a drug which works, which has high response rate. It works why, and does it work in all populations, and does it work well in those populations? Lots of questions need to be answered. It's not very simple that you got a new drug and then you chuck it in everybody. I talked a little bit about the family history and the small increase in likelihood, and it is a small increase. It's very much an area of research interest for us. This observation stemmed from the fact that we're finding more MGUS around the index patient with myeloma. Where Simon works in Royal Marsden Hospital, there are some research groups who have taken samples from myeloma patients and controls, and they've looked at what genes may be driving this change. And the interesting thing is it may not be just one gene, but it may be several sets of genes. And there is a word that you'll have to get used to, and it'll come through in even public language in the next 5-10 years. It's called polygenic inheritance, which is basically, it's not a single gene in your body which decides whether you have the disease or not. It's actually more than one gene, okay? So it's called polygenic inheritance. And there are going to be panels being made available. There are many companies in Oxford and Cambridge who are making these panels, which will probably be made available. Risk of diabetes, risk of this, risk of that, you could probably compute. So we think that it's not a single gene, it's probably going to be polygenic, which makes it even more complicated as to how you develop those tests and assays is what it is. And the most recent research that has happened in this space looked at like how you would excavate some place and get an object and carbon date it. We've been carbon dating the DNA of myeloma patients, and when you carbon date those DNA, you find that DNA change may have happened in their late teens or early 20s. So there is a school of thought that now tells us that if MGUS were to develop in a person, it probably develops their late teens or early 20s. So one of the reasons when there was an active conversation this morning and the age was mentioned, I was a bit concerned because the answer is, if you test too early, then you wouldn't know that they've developed it, and we just don't know when the right test is. So there's a lot that we need to know before we start to really get involved. So, so all of those are quite interesting for us to think about and be thinking through that. But the simple question, and this question I get from myeloma patients who I diagnose, and my clinics are on Friday afternoons, and I was diagnosing somebody yesterday, and I got the same question as to why have I developed myeloma. And the answer really is we're continuing to research. There is no single factor. And everything that you can think about, pesticide use, we've researched. Hiroshima, Nagasaki, everybody who's been lived around the epicenter has been bled, and MGUS tests have been done. So anything you can think of, we have done that research. And we can't find anything. We're just finding associations. And the problem is what I tried to explain to you earlier, because we always categorize. As doctors, you know, if I take myself to 20, 30 years ago, I decided what myeloma definition should be, right? Okay, so therefore, there is a bit of challenge in trying to attribute everything to myeloma itself. And nobody else, you know, if you then attribute to smoldering myeloma, then your association becomes significant. So those who understand statistics better know that when you've got a larger pool of population, then your attribution becomes a lot more stronger. So we are struggling with those challenges, is what it is. But I think the way I'm going to summarize this is to say, potentially what is happening is there is something internal within the genome which gives you a predisposition, and then there's something happens during living life which kind of makes that change to happen. So it's, as how people describe, called the double hit theory, okay? One hit is already here, and you need to get the next hit to have a problem, is what it is. So that's why risk cannot be computed that easily, okay? Well, if you were confused before, you'll probably now be more confused with all my explanation, okay? Anyway, I've tried my best. So, as I said, how does myeloma start? It starts in the genome. A normal plasma cell has nice pairs of chromosomes, which are all equally arranged and so on. And in the MGUS stage, you start to develop some genomic changes. So when those genetic changes happen, then those cells are immortalized. So they don't die, or they have a small turnover, and you will always maintain that clone in that bone marrow over a longer period of time. And the only observation that's quite interesting is when people with MGUS get quite severe infections, sometimes the MGUS clone just dies. And that's because they have such a huge T cell response against that infection, and then the MGUS clone just dies. That's been the only observation, which was partly the reason why CAR T cells were developed, and they were hoped that they would make a huge difference in myeloma. Even an MGUS clone can die in those people. But having said all of this about the precursor states, and having made a case that we should be doing research in the precursor state, thankfully still, by the definition of how we call multiple myeloma today, very few people get to multiple myeloma, okay? That still remains the case. We have not observed, just because we have more testing these days, that more people are actively moving on from MGUS to myeloma. I get asked these questions, even in academic chambers, as to, are there more myeloma? I don't think there is more myeloma. We're just doing more tests, and we're picking up more myeloma. And more people are aging, and the projection in one of the estimates that I've seen is that there'll be almost double the number of newly diagnosed myeloma patients as defined today as what is myeloma, you know, fractures, anemia, and so on, in 2035 is what it is. And that is a feature of just more people aging and more testing happening in people with myeloma. So the first thing that I always explain to people that, you know, people talk about screening programs. The interesting thing is, if you go out and screen today, you're going to screen more people with myeloma. You're not going to screen people with MGUS. You're just going to get 30, 40,000 people with active myeloma sitting out there in the community who've got a bit of pain here and a bit of pain here who will actually be picked up with active myeloma. That's the biggest impact of screening program. The benefits are almost immediate when you actually do a screening program. It's not much later. You'll sweep up a lot of people. But our NHS can't cope with 25,000 patients, leave alone 40,000 newly diagnosed patients, right? I mean, how are we going to get them all transplanted in that 1 year? So there are some distinct challenges. Sorry, I digressed a bit, but that's where it is, okay? So as you can see here, genetic changes are not immortal. Unfortunately, the plasma cell is immortal, but the genetic changes are not immortal. They keep changing every year, every month, and what treatment you give. And that is part of the challenge. So you create yourself more challenge by more treatment. So that's also something to think. And we have signatures created by the drugs on the myeloma cell as well, which then becomes harder to treat down the line. So an ideal situation, as you can see, I'm a big proponent of treating early and treating smoldering myeloma. Treat aggressively early on, but stop treating. And then you just keep the clone in a quieter. What happens with continuous treatment? There was a question this morning about how long you use lenalidomide for. If you think through that, then one of the concerns you have is you're continuing to change the DNA within those residual plasma cells, and you're creating a rod for yourself. It's what it is. Honestly, I'm not giving a message you should stop lenalidomide. You should continue lenalidomide if you're on it. But you can see the theory that you can think about. So it's important to recognize that myeloma genome is not immortalized. It continues to change with time. And Simon talked about it this morning as well. So part of the incurability is partly created by the way we think about treating myeloma today. And that has to change over time. We may have to think hard-hitting and then give people lots of breaks and therefore let myeloma germinate and become more vulnerable like how it was when we first treated it, right? Lots of different ways of kind of thinking about it. So these are things that we are testing currently in research as to how we can do that. And there are several different subtypes of myeloma, and it's very important to think about that because Myeloma UK as an organization has had a deep interest in this because it's always looked at the unfairness in which every myeloma patient is getting the same treatment, and that's not fair. And that's not fair because you don't get the same responses. And if you don't get the same responses, then you should think about how you treat them differently. And the different genetic subtypes means different outcomes. And that's why MUC9B was a trial that was, Myeloma UK 9B was a trial that was led by Martin Kaiser and Matt Jenner. And we now have proof from the clinical trials that we have run that giving a very differentiated treatment based on the genetic subtype of myeloma makes a significant difference to the outcome of people. So part of our lobbying, as we continue to do with NHS England, is that yes, new treatments are getting better and better. We are excited about that, but we need a bit more flexibility around with the new evidence available to put the treatment program together differently for people with certain genetic subtypes is what we want to do. We want that flexibility. NICE come back to and says, give us the evidence and get the drug companies to come and present evidence. But drug companies are never going to do a separate treatment program for high-risk myeloma and non-high-risk myeloma. They just wouldn't because it would take much longer to do. They'll have to make a lot more investments and therefore they are less interested. What they want is the bigger group of people, and obviously there is a cash value at the end of that, and they need to be able to demonstrate that it works for everybody is what their interest is. So it still considerably concerns us that the outcomes are different, but we are using the same treatment for all patients. In the precursor space, Myeloma UK has stuck its neck out in funding some of this precursor research. Some of the ideas that I was sharing with you earlier on. So Chris Bunce is based in Birmingham, and he's thinking about, well, if we're not doing a screening program today to try and find that 1.5 million people with MGUS because we're worried that we'll be worrying them, or we wouldn't know what to do with them, and suddenly we have 1.5 million people we have to monitor in the hospital. If that's the NHS concern, then how do we actually ask certain questions which can then help us address those concerns? So he has taken some basic health economics around currently diagnosed MGUS patients and how they're converting to myeloma and what is really happening in them. But he's also doing some laboratory research to try and understand as to whether we can use some currently available treatment that can be repurposed to keep this MGUS clone in an MGUS state and not let them even convert into smoldering myeloma. So that's what he's currently doing, and we're proud that we've funded him, and he's now gone on to get some additional funding from Cancer Research UK. And why are we excited? There is a big screening program currently in Iceland. I don't know if you've heard of it. Some of you may have. Whole of Iceland has been screened for MGUS. Anybody over the age of 40 was invited to take part in an MGUS screening program. 60% of the population put their hands up, so we have done that, and all of those samples are going to be analyzed in Chris Bunce's lab for some of the laboratory research that he's going to do. So it's hugely helpful. Is it going to give us all the answers? No, because as you know, Iceland is a very genomically homogeneous population, and therefore we need answers from other areas as well. So there are some other research happening as well. Kui Yong at UCL, some of you may have heard the study that I've got in the next slide, is looking at smoldering myeloma and looking at how you stop smoldering myeloma converting to myeloma as to why it happens, and then look at treatments to prevent this from happening. I've just complained to you that NHS England doesn't give us the flexibility of using different treatments for different people. Having made that complaint, we should not be complacent. So we're thinking, well, if we're making that complaint, then we need to be doing that research to show that smoldering myeloma don't treat everybody the same. Treat some people in this manner, treat some people in this manner, and therefore you get the best outcome. So we are developing a data set to look at in whom where the immune function drops, and that's why they get active myeloma, and in whom where the genomic change develops, and that's why they get from smoldering myeloma to myeloma, because the treatment that we would give for these two populations will be very different. You can immunomodulate with lenalidomide for patients who are just losing immune function, whereas in whom, where you're developing more mutations in the myeloma cell, as you go from smoldering to active, you need active treatment to kill those cells. You can't immunomodulate. It's beyond that immunomodulation, so you need combination treatment. So that's how we're thinking. So we're thinking about whether you have different prescription for different people as you move from smoldering myeloma to active myeloma, okay? So as I said, we can't complain, we have to make, new treatment programs for people. This is a study, this is open in about 30 odd hospitals called Cosmos, and a number of people have put their hands up for this study. We have over 300 patients, and I'm pleased to say this is the largest study ever in smoldering myeloma, observational study. I'm pretty sure worldwide. There are a couple of studies in the US which are looking at smoldering myeloma, but nothing as rich as this. So we should congratulate ourselves and continue to put more patients. Professor Kui Yong and I and myself are writing a grant which goes in next week so that we can get more money to keep this going, so that we can get 600, 700 patients, so we can study their biology and as to what is happening from smoldering to active myeloma. So a lot of what I've tried to do in the kind of first section is really describe that a number of you would have active myeloma. Typically, that's expected because that's how you associate yourself with Myeloma UK. But there is a whole precursor piece that's going on that our focus is shifting. And if that comes through in some of our Myeloma UK information, you now understand why we should be focusing on the precursor program, because it's critically important we get answers from that patient population so that we can move even treatments along in active myeloma. So having talked about the whole precursor space and how myeloma develops and what may be going on, I'm going to jump straight into treatments and how do we tailor treatments. I've already talked about the fact that we do it based on genetics, which is right up there, as you can see, which drives patient outcomes. But now, as we have large data sets, we can now look at what are the things that drive patient outcomes. And one other thing, as I said, about 45% of people diagnosed in the UK are over 75, right? Okay? So more people are frail as we see them in clinic. So what does frail mean? Frail means weak, okay? But I would say weak enough to not get the 4 drug combinations and 3 drug combinations that we would just dish out to people, right? We will just prescribe those treatments. And that can be taken by a 60-odd-year-old person who has got probably a bit of hypertension and is on a blood pressure tablet. But if you've got somebody who is 76 and has got atrial fibrillation, osteoporosis and hypertension and diabetes and a bit of poor kidney function, and if you give them the same treatment, they're going to feel even more frailer, right? So there are trials, as I'm going to show you, where we are looking at how we look at frailty as a stratification factor. And we're looking at response to treatment early on. So this is what I call as dynamic way, okay? So a person walks in, you look at frailty, you look at genetics, but then you also continue to look at the same person at different time points and then look at how have they responded to this treatment and whether we can change their treatment based on how they responded. So you don't just stratify when they first walk through the door, you stratify after a few months saying, well, having done all of that, actually we think, Mr. Smith, your best treatment from now on would probably be this. So we want to be able to tailor those treatments and immune biomarkers. So I've been repeating the word stratified medicine. So essentially what we want to do or get into is getting into giving treatment based on individual factors that we see in individual people, rather than telling you, oh, we've got this new combination licensed and this is what's funded by NICE and we should give you this because that's what we would do. Well, hopefully the language will turn into, Mr. Smith, because you've got this particular genome and because you've had these challenges, what we're going to do is going to try this treatment because this has got the highest chance of making you better. But we're not going to stop there. We're going to remeasure in about 3 months, and we would like you to be in this place because if you're not in this place, then we're going to do this. So that gives you the confidence that you're not just one among the 6,200 people who are diagnosed with myeloma in the UK every year. You are an individual and you being cared for appropriately with the right set of data that can be captured in your clinic. So that's really what stratified medicine is. The most important piece is this, because everybody's going to get side effects, okay? That's part of taking even, dare I say, paracetamol or amoxicillin, You would get some side effects from it. But how can you get away with the least set of side effects is what you need to think. And for the least amount of time is how you need to think about it, because it's all part of the package. Good comes with the bad, but can we limit the bad as much as possible for people? And you could do that better when you do stratified medicine, where you're thinking about the person in front of you rather than this is myeloma and this is what we do. So we've got to kind of move away from that. So these are the trials which I can proudly claim are all happening in the UK. It's very interesting. Very few countries outside of the UK do stratified medicine. I'm looking at my colleagues here, medical colleagues here. I'm sure if you can think of all the trials that's happening, I mean, how many of them are stratified to any of these? Everybody would want to just look at next new drug plus next new drug is what they would look at. In the UK, we're proud to do these type of trials, and we also do antibiotic trials. What Stella led and showed the evidence that bringing in antibiotics for the first couple of months makes a difference to patient population. So that's what we're very, very good at. So there is something that in the UK as a research community, we continue to contribute, and it's very, very unique, and that's what this does. So I've already said about the MUC9 that was specifically looking for genetic high-risk patient population. We have now shown that there's a particular treatment pathway which works, and Myeloma UK is working with the chief investigators and made a submission to NHS England to say, can you please consider a separate package for patients with high-risk disease, is what we've asked, and it's under evaluation right now. The RADAR trial is currently active. We're looking to recruit 1,400 people. And for the first time, we're asking the question, I think, again, this morning we spoke about continuous maintenance. We want to stop maintenance treatment. We don't want to be giving treatment forever. And we're hoping to achieve that in about a third of myeloma patients who are newly diagnosed. So it's the first trial in a randomized fashion which is going to stop treatment for myeloma patients after a period of time. But we're going to do it in a randomized way, which means we'll find the exact truth, as much as the exact truth can be in this scenario, okay? And then the fitness trial is based on initial fitness. As I said, everybody gets the same treatment when they come in. And the only treatment that gets attenuated or reduces steroids that you've just heard about previously in a really nice talk. That's the only treatment we currently reduce when patients come into hospital. We're now looking at reducing all treatments that we would give for newly diagnosed myeloma patients based on the fitness. And is that a safe thing to do? And does it mean you get a better outcome is what we want to study. And that's what we're studying in the fitness trial. So really, really good research that's happening in the UK in all of these areas, and hopefully will inform. So even though these will result in, whatever the outcomes, and even in the next 5 years, you still had a straitjacketed, one-size-fits-all type approach, we will use information from these trials to still personalize care of patients within that straitjacketed treatment. Are you with me? Okay? So we may not have a licensed pathway to stop treatment early and whatever, but we will use inferences from these trials to actually drive care for patients, because these are all learnt by your doctors who are looking after yourself. I mean, a number of these trials are running. I mean, we have about 200 odd NHS trusts. These trials run in about 80 to 100 hospitals. So, if you were to bump into a hospital, they'll be running, one of these trials or all of these trials is what it is. So that is about having current treatments that we have and how do you actually tailor them. But developing new treatments is critically important in the field of multiple myeloma. This slide was shared by Simon earlier on, and a lot of work is happening in the laboratory. More and more mute targets are identified. More and more phase 1 trials are happening in myeloma and getting into licensing. I think in the last 5 years, there's been like 13 or 14 drugs approved in myeloma, and myeloma is the single most indication which has had more FDA approvals in cancer than any other cancer. So that tells you the wealth of research that is happening. I often cynically say, I hope there's no pharmaceutical company folks here in this room. I don't know if you've been, again, I'm digressing, but I'll say this anyway. If you've seen a pattern, every major pharmaceutical company has now got an oncology arm, right? Novartis Oncology, and you've got Pfizer, and everybody's got an oncology arm. The reason for that is everybody's figured out that having cancer treatment is the best way of making money. You can't make money by selling baby powders and whatever. There is a problem with some of these, okay? So they've all got oncology drugs. And within oncology, what is the drug you can make most money on? Multiple myeloma. So don't be surprised. Every major top 10 pharma company will have a myeloma drug. If they don't have one, they're trying to find one, okay? So that's exactly what's happening. So the pipeline is massively rich. Okay, and more people are investing. I only know of 2-3 other pharma. If their drug is successful, then all the top pharma companies will have a myeloma drug, and they'll all be fighting against each other, which is what NHS England want, because they want to drive down the prices. Okay, sorry, I digressed. But anyway, that's the point. Okay, so but all of this goes through NICE and then gets adopted into the clinic. But what we really need to drive is the whole kind of patient needs and preferences as well. So these trials, it's so nice to see so many of you taking part in these clinical trials. And I think it was Simon who said earlier, and that's an important point, do not be worried about not getting CAR-T. You could probably get CAR-T 5 years earlier if you participate in a clinical trial. So put your hands up. Always think about, you may have to travel a little while. Again, Simon alluded to that. It may not be convenient, but, participate in all the clinical trials. So many pharmaceutical companies are coming and doing clinical trials, in our country. Always ask the question, is a clinical trial available probably 30, 40 miles away, or probably in your hospital, which will give you a better, treatment. So there's always those options available. And within Myeloma UK, we're continually thinking about how we facilitate more and more of these trials to come into the UK. So phase 1 trials is those early phases where treatment is typically looked at. It unfortunately typically tends to happen at the Marsden, Oxford, King's, Christie, or Leeds, unfortunately. They seem to be the centers where it goes to. But we have to, hopefully Myeloma UK has to campaign and diversify this and get out to other geographies, because I think it's a bit unfair. And phase 3 trials are much larger, and that's what gets into 80 to 100 hospitals around the country, which is great. So how could you take part in your clinical trial? As I said, not everybody will be suitable. It may not be the right trial, or it may be a 1:1 randomization to a current standard of care which your doctor does not want to give or would not recommend to you. So there are many reasons not to do a clinical trial, but there may be many reasons why that will be in your best interest. Unless you ask, unless you email someone or pick up the phone and speak to a Myeloma UK helpline, you're not going to find out. So that's what I'm encouraging you to do. You can be very confident. They go through a strict ethics committee process. I can tell you it's the strictest out of all Western countries. I mean, I'm amazed what people are able to get away with when they write research protocols in America. I won't be able to get away with that in the UK, because I know what questions I'll be asked. So you can be confident that whatever's presented to you has had the rigorous way of being checked. What happens from an operational perspective, you get given something called a patient information sheet, which has got the language about the trial as to why it's done, how the treatment's given, all of the different side effects, and what are all the different tests that are needed. As Simon alluded to, sometimes it can be a bit labor intensive, and you've got to take that into consideration as well. So you need to have that candid conversation with your doctor saying, do you really think I should do this? Because it could mean like 3 years of commitment to. So there may be good reasons why to not take part, because you've got caring responsibility and so on. But there may be a good reason to take part. So important thing is to read the information sheet, talk to your doctor and nurse specialist and your research nurse. And typically there's a waiting period before the whole initial screening starts, and then whatever happens downstream. And clearly there are folks in the room who've been through that pathway. So this is how drugs are developed. So from the lab all the way through the clinical trials, and then they start to get licensed and approved. I've got 3 or 4 slides to talk about how the licensing happens. So it's the only time I'm going to mention the word Brexit. Okay, we have done a Brexit, and therefore previously we used to have an EMA approval, and when EMA approval happened, we used to have drugs immediately come through to the UK. But drug companies now have to take a separate dossier to MHRA, and that's how drugs are licensed. Okay, so when you start to Google, or if Myeloma UK sends you an information leaflet saying EMA has approved this drug, that doesn't mean it's available in the UK. UK has to do a separate submission to MHRA. Okay, so that's how it is. So that's called licensing process, which basically means it's a safe treatment program to be considered for myeloma. That's what MHRA say. But then there's things happen after that, which is approving treatments for the use in the NHS. So just because a drug is licensed doesn't mean NHS is going to offer it to you, because the drug comes with an MRP, and that MRP is unacceptable to the NHS. So we need to get the MRP, we need to get a discount on the MRP to get it right for the NHS, okay? So what we need to do is we need to do a health technology assessment, okay? So we ask the drug companies to present to us data on how effective the drug is, how does it improve the quality of life, how long people live when they take this drug in comparison to what we can give now, and therefore attribute a value that we are prepared to give, okay? And that is in health technology terms called QALYs. And we want a certain level of QALY improvement, and we are prepared to pay a certain price for that QALY improvement. So that's what happens here. NICE does all of that work. In Scotland, it's the Scottish Medicines Consortium. And this is where we need a lot more influence from myeloma patients, and we really need more of your voice, as there's more and more public consultation about combination treatments and diagnostics in myeloma and so on. please put yourself forward and feed in your comments. And if there are ideas that you have, and if you want to join the advocacy partner panel, please do so, because you will essentially be feeding into NICE and NHS England, and they will look at all of this, because there's always, so the drug companies will always try and get to a certain price, probably about kind of 5% above the threshold, and NICE will sit there debating forever as to whether it's worth paying that extra 5%, okay? But if the patient language, so this is what I always tell when I advise drug companies, I tell them, okay, the power in patients and us is, if we say no, NICE immediately say no to you, because we don't want it, you won't get it. But if we say yes, we need to improve our language to say why doctors and patients want this drug is what we need to do, okay? So we have the power of saying no, because that means less money spent by the NHS. So that's why they ask us whether you need this treatment or you need this new test or whatever. We always have the power of saying no. If we say no, everything gets stopped. But if we say yes, we need to color that in. And we have to say why we need this drug is what we need to do. And for that, more of you have to get involved. And you will then see the difference. And some of the stuff that we discussed this morning about CAR T. We could always write a paper together and ask government the question, how can we actually get CAR T? It's unacceptable when it's available in the whole of Europe that we can't have access to CAR T. Think about it that way. A bit of revolution is what we need within the community. So research and new myeloma treatments. So what I want to give you is some examples of how we've made a difference with the research that's happened in the UK. I don't expect you all to know the Myeloma 9 trial, which happened in the early 2000s, which looked at actually thalidomide and dexamethasone combination. But we used zoledronic acid for the first time in the early 2000s and compared it against a tablet called clodronate and wanted to show where that zoledronic acid was better. And the data that was collected on zoledronic acid made an international influence of use of zoledronic acid. So those patients in the UK, and there's about 800 of them who participated in the early 2000s having zoledronic acid, is why everybody in the world currently gets zoledronic acid. A powerful message, and that's the truth. It was published in Lancet and made a huge difference. Second example of why research makes a difference is the lenalidomide maintenance, and some of you may be part of the Myeloma 11 study, and lenalidomide maintenance is part of that study. You may say, well, lenalidomide maintenance was even available before, but lenalidomide maintenance is given 3 weeks on, 1 week off. Before that, lenalidomide maintenance was continuous every day, and the difference was made by the Myeloma 11 trial. And we now have the ability to give lenalidomide maintenance because of the Myeloma 11 study. So some really kind of powerful examples of what difference we can make to patients. So I've talked about how research can improve new treatment. I'm going to finish up with a couple of slides on the brand new treatments that are coming through, which are the bispecific antibodies and the CAR T cells. Really exciting. And there are also, I shouldn't ignore, there are other trials that are opening up or running right now, iberdomide trials. So you've got lenalidomide, pomalidomide. In that sequence, we now have iberdomide and mezigdomide. So newer drugs, more effective they are. So they will be made available to you in clinical trials early on. And then we've got the antibody drug conjugate, belantamab mafodotin, made by GSK. That drug in combination is extremely effective. There are 2 indications going through NICE, and we're really excited to use this treatment combination. So it's not just bispecifics we're excited about. We're excited about all of these new treatments that are coming through. CAR T cells, for those of you who don't know CAR T cells is essentially a living drug. We take your T cell, send it to the company. The company puts a T cell receptor in, which can recognize a myeloma cell in your body. And then they send back the pack, and we give you a 100 ml pack of blood, and that contains the engineered your own T cell. And those T cells go straight to the myeloma cell and kill them instantaneously. They tend to live in your body only for about 6 months, and then they die. And so you get that instant hit, and then the myeloma stays in control. Unfortunately, in leukemia, we saw cures in kids with CAR T cells when it worked. In myeloma still comes back after about a period of time, and it tends to be when you use it in a myeloma patient. If you use it much, much later, it's about a couple of years. If you use it much earlier, it could be 4 years or even longer is what we're expecting to see. So that's CAR T cells. And the final thing to say about CAR T cells is not all CAR T cells are the same. Different companies have different CAR T cells because they could be targeting different protein on the myeloma cells. So don't think every CAR T cell is the same. Every CAR T cell will be different in how they're made and how they're engineered and so on. So massive excitement around CAR T cells. But bispecific antibodies is what we've got first our hands on. And this is the exciting new way. So we've always had antibodies to treat blood cancers, and then now we have got antibodies to treat breast cancers as well. And now this is a clever way of making an antibody. You've got an antibody which sticks to a myeloma cell, but also sticks to your body's T cells at the same time, fairly instantaneously. And it brings the T cell closer to the myeloma cell and kills the myeloma cell. So really exciting. And the protein that it can attack on the myeloma cell can be different. And we've got 2 bispecific antibodies licensed against the same protein called BCMA. But then there are newer antibodies coming through which will target different proteins on the myeloma cell. So if one fails, you could try another bispecific antibody. And even that fails, then you could probably go to another bispecific antibody. And a lot of that's coming through. And this is what I meant about all the big pharma. Everybody's having a bispecific antibody against a different protein, so that they can all get into the field is what's happening right now. So these are the exciting new drugs. And obviously, again, one question this morning about why don't we use bispecific antibodies early is what it is. It is happening. I use bispecific antibodies for newly diagnosed patients in clinical trials. And as you would expect, we see some profound responses early on in patients. But all drugs have side effects, and there are problems with bispecific antibodies as well, as you will hear and you will realize in due course. So always having myeloma in remission, not having drugs is the best place to be if we can get there. More information about new treatments and clinical trials are available in our Myeloma UK web pages, as you can see here. And information about new immunotherapy and the clinical trials are all here, as you can see. And there's information about belantamab, CAR T cells, and also bispecific antibodies as well. So I've tried to cover the research and why we're researching certain areas in myeloma. I've tried to cover how clinical trials are run and how the treatment approval process and how you could get involved. Hopefully, I've covered the main messages, which is research is critical and underpins our ability to continue to improve care and life chances of people. If we didn't do research, then we wouldn't have got to where we've got to. And it's all thanks to a number of you who are part of it, and hopefully a number of you can continue to support it. And there's plenty of new treatments coming through and new treatment strategies, and we will stratify them in due course. We won't tell you just because one trial said it, we're going to do that. We will have more information to personalize care for people. If that conversation is not happening, you should initiate that conversation. It's very important that you drive that. And your involvement is absolutely critical. So I'll finish up with the Myeloma UK slide, and thank you for putting up with me for 50 minutes. Any questions?
Hi, yeah, I've taken part in several clinical trials, but the big problem is you take part in a clinical trial and you never seem to find out what the result of the clinical trial is. So, if you want us to, add to patient advocacy, actually getting the result of what's happened in the clinical trial would be quite useful.
Absolutely. So I think that's a message. I think Myeloma UK as an organization, we can probably take that back because we know what clinical trials are run in this country, and we could probably get the key messages sent out to people. We can work with the chief investigators to do that. So, critically important. Yeah, absolutely. We have to get that right.
Hello. Is there an age limit to undertake the CAR-T
There
treatment?
isn't, and I was being very careful when the question was asked this morning because it's a younger. I think you could do. I mean, there are people who are 83, 84 who've had CAR-T already. So I don't really see it's an age-specific treatment, but it's a fitness-specific treatment, is what I would say. So I don't think it's very age-specific. And so what is the concern about CAR-T? The concern about CAR-T is the cytokine release syndrome when the T cell meets the myeloma cell in a person's body, okay? So the newer trials are looking at giving the standard treatments and getting the myeloma down, and then giving the CAR-T cells as what we use the word consolidation to keep myeloma in remission for longer. So when you do that, why would you want to differentiate whether they are 85 or whether they're 35? They should all be able to get CAR-T cells. Are you with me? But the T cell fitness may be different. But we currently don't have the test to decide whether a person's T cell is fit enough to be taken as a CAR-T cell, if you understand what I mean. But when you send T cells to the drug company, some of them, so there are 3 results that you can get. Very good cell. You've got a good product, go ahead. Suboptimal product, you have to decide between you and the patient whether you will give the T cells, or you'll get an email saying, I'm so sorry, there's no T cells coming because we couldn't manufacture. So you'll have 3 results coming back. So that's why I want to give people the message, CAR-T cells are not everything, because there are many reasons why it may not be the right thing. But I don't think it's age-specific based on what all is currently happening. But when it first came out, there was a feeling that it will only be for younger patients. I don't think that's the case. I'm sure if there was another myeloma doctor, they'll argue, no, absolutely no, it should be less than 60. But I don't think so.
Can you harvest them early on and treat from the stem cells?
Yeah, who's going to pay for it? So that's what we're currently thinking through. We're already filling up our NHS-BT tanks with stem cells, and they want to check it out, and we're trying to stop them from checking it out. So if we get T cells and store it down, then how's it going to work, and so on. Absolutely, yeah. Those are things we're thinking about right now, and we don't have answers. There is, University of Pennsylvania is very, very good. That's where most of the CAR-T cells were initially developed, and they've already shown that if you do that earlier on, then it makes a difference for people. So we're thinking about that, but it would mean a huge infrastructure in the UK to do that. Can you imagine? And when would you collect it? If somebody is currently on lenalidomide, would you stop the lenalidomide and collect it? Or would you leave them free and, would you get a different T cell if you're on lenalidomide or if you're on daratumumab? There's so many questions, right? So it's not quite easy. So first thing is to land, yes, it's always useful to collect it early. Once you've landed that, then you've got to work out how do you do that, when do you do that, and what are the resources that may be required. But there will be big pushback from NHS England if you wanted to do that.
If we're moving towards personalised treatment, and as the Optimum trial showed, myeloma isn't one disease, it's a series of different diseases or subtypes of disease, how are we going to do that without more genetic testing and being tested at relapse as a norm, as opposed to that being done within clinical trials? But as a patient, it's very difficult to get genetic testing at any other time other than at diagnosis. And there are some patients who are still not being given FISH testing at diagnosis in some hospitals on the basis that treatment is the same irrespective of the results, and yet the treatment can't be differentiated unless different subtypes of myeloma are identified. And at the moment, we're having perhaps many patients having treatment which doesn't stand a chance of working because of their subtype of myeloma, because we're all going through the same, well, perhaps not exactly, they're certainly the same induction treatment. I mean, it was good to see on the slide this morning how many different options there are for second-line treatment. But anyway.
Yeah, absolutely. So all relevant points you've made. So we do need to make progress on that. But as you saw on the list this morning, I think more options are being made available and more newer options will come through for newly diagnosed patients. So doing genetics, there'll be a more of a reason. Everybody's looking for a reason to do the test, because if you talk to doctors, they'll say, oh, resources and it's painful for patients, and that's the reason why they have a system operating within their hospital. So the biggest driver is we change treatment based on what the genetics is, and there are more options being made available, and that will drive treatment. I think one of the things that I don't know if it's been discussed this morning is whether or not you do stem cell transplants may be based on genetics in the future. So we will differentiate care based on genetic makeup of myeloma. And the second point you made about doing it around relapse, that's the recommendation, as Simon said. We are recommending that people do genetic tests even at relapse, because myeloma cell is immortal, but myeloma genome is not immortal, and genetics changes over time in patients. So the recommendation is to do that. And you again have lots of different options for patients based on genetics. They may not be the trials that was run like the MUC9B specifically for high risk, but there is evidence that if you use this treatment combination, this is shown to be more effective in patients with high-risk genetics, is what's coming through even with the current trials. So your doctors should be able to pick treatment at different relapses based on what the genetic subtype looks like. So yeah, I think we have to continue as Myeloma UK organization, and BSH guidelines should continue to impress upon people to do the genetics of myeloma at every stage of myeloma.
Brilliant.
Thank you.
Thanks.